All right. Our second speaker this afternoon is Dr. Caroline Strömberg. Dr. Strömberg earned her PhD from the University of California, Berkeley, where she studied tertiary grass-dominated ecosystems. She did postdoctoral research at the Swedish Museum of Natural History, as well as the Smithsonian National Museum of Natural History. For nearly 2 decades, she has been a professor and curator at the University of Washington, where she uses fossils to explore evolutionary and ecological links between plants and the environment. Today, she'll speak on the deep and tangled evolutionary roots of North American grasslands, insights from the fossil record.
Thank you very much. I'm sorry that I'll be keeping you from coffee a little bit longer. So I'm very excited to be here. And let's see, let me see if I can use this. Okay. So I'm excited to be here, and I am a paleobotanist. So I'm going to talk about some different kinds of discoveries in the fossil record that we can use to reconstruct the evolution of North American grasslands. So let's see if this works. Okay, so first I wanted to remind you just how important the North American grasslands are. Grasslands make up about 15% of the continent as a whole, and of the US, 40% of the land surface. They host important biodiversity. They are the basis for our agriculture, and they also are important for carbon storage, in particular underground. And they are important for filtering water and also for stabilizing soils. So grasslands are super important in North America, but also very threatened. So over half of the area of natural grasslands have been converted, for example, for cropland, and less than 9% of the remaining grasslands have any kind of formal protection. So very important, but very threatened. And because they are so important, paleontologists have been preoccupied for about 150 years in trying to understand when and how these ecosystems came about and why. So I'm going to be talking about some work looking at that. But first, I wanted to just give a brief sort of recap on how we can study grass evolution and grassland evolution in the past. So paleobotanists have of course tried to probe the fossil record of grasses, but unfortunately it turns out that this fossil record is a little bit ambiguous, in particular in North America. So grass pollen, for example, have traditionally not been very useful within grasses because grass pollen are fairly uniform, although I will say that the work in the last 5 to 10 years has really started to break this open and make pollen much more diagnostic within the grass family, Poaceae. Also within the Great Plains, where many of the grasslands we have today are, there are just very little pollen preserved in the fossil record. Grass leaves are practically useless unless you have anatomy preserved, which we almost never have. Grass fruits, however, so diaspores, have very diagnostic traits, and they have been studied in the Great Plains, so Nebraska, Kansas, Colorado, for about 100 years with Max Elias' work and Joe Thomason's work. However, these fossils remain rare until about 10 million years ago, so not super great if you want to look at deeper time history. So what people have done instead is rely on indirect or proxy evidence for past grasslands. And in particular, people have looked at, or paleontologists have looked at the functional morphology of large herbivores, or many cases ungulates. And in particular, focusing on the evolution of low-crowned or brachydont. Oh my gosh. Teeth? No, you can't see it. On the left, such as a human tooth, to high-crowned or hypsodont cheek teeth, such as on the right. And this is a tooth of a horse, and you can see these hypsodont teeth in the jaws of that horse. And so when you look today, you see that hypsodont herbivores tend to be concentrated in grasslands in terms of their habitats. And people have thought that this was because these really tall cheek teeth allows the teeth to be worn down in abrasive feeding, which happens in grasslands, either because animals are feeding on silica-rich grasses, or they get a lot of soil and dust in their mouths as they're feeding really low to the ground. And so the assumption has been traditionally that Hypsodonty evolved as an adaptation to feeding in grasslands. And paleontologists have used the appearance of hypsodont animals in the fossil record as a marker for when grasslands spread. So here you can see in North America, hypsodont horses and other animals started evolving, or some of them at least, a large number, around 18 to 15 million years ago. So that was traditionally the thought, the timing of the spread of grasslands. But in the last 25 years or so, people have started using an alternative line of paleobotanical evidence in the form of phytoliths. So phytoliths are microscopic silica bodies that form when plants take up monosilicic acid through their roots with the water and then deposit it in and around their cells in the living tissue. And this is an example of a phytolith in the epidermis of a grass. And then once the plant dies and their tissues decay, these phytoliths are left in the soil and can provide information about the plants that grew at a site. Phytoliths occur across land plants, but they are particularly abundantly produced in, for example, horsetails, palms, and grasses. The taxonomic specificity of phytoliths varies among taxa, so they're not very good at providing a detailed list of trees and shrubs growing at a site, but they have been shown to provide useful information about sort of the broad vegetation type, so whether you're looking at a savanna or a forest. Also, within the grass family, Poaceae, they are taxonomically quite variable, so useful. And for this reason, this is a phylogeny, by the way, of the grass family. We can use them to separate different major taxonomic groups within the grasses that are also ecologically important. So for example, early diverging grasses, bamboos, and rhizome relatives in the Oryzoideae are ancestrally adapted to closed habitats, wet habitats. And then two clades independently evolve the ability to thrive in open, drier habitats. So first, the cool temperate poets, which dominate temperate grasslands. And then the so-called PACMAD clade, which contain tropically adapted open habitat grasses and also closed habitat grasses. And within the PACMADs, we see the independent evolution multiple times of C4 photosynthesis. And the C4 grasses are what dominate tropical grasslands and savannas today. So phytoliths are useful for studying the diversification of ancient grass lineages and also looking at the ecology of past grass communities. So I'm going to talk about what we know about the evolution of North American grasslands. When did open habitat grasses originate, diversify, and arrive in North America? When did C3 and C4 open habitat grasses become ecologically important? And a little bit about potentially why. And then I want to talk about how similar the earliest grasslands that we see are to modern North American grasslands. All right, so before I go any further, I just want to acknowledge that this is the work of many, many people, including my own, grad students, postdocs, etc., and many other colleagues around the world. Okay, so let's first talk about what we know about the evolution of grasses writ large. So for a long time, we had pollen evidence that suggested that grasses had evolved at least, or originated by the latest Cretaceous or early Paleocene. And these pollen were all confined to the southern continent or Gondwana. So that's where people assumed that they originated. Phytolith records from the last 20 years or so have supported an early radiation and pushed it back even further to maybe even the early Cretaceous, and also expanded the biogeographic range with these fossils from China. Nevertheless, when you use these fossils to put them in a biogeographic analysis, in a sort of using the phylogeny of grasses, we still end up with a Western Gondwanan origin, so maybe South America or Africa. Also, these reconstructions show that most major grass clades probably diversified outside of North America initially. And so they must have immigrated to the continent in the last 66 million years or so. And then if we look at open habitat grasses, so poets and packmats, fossil phytoliths, and a recently described spikelet, point to an early origin of open habitat grasses, so in the Eocene or something like that. And this holds true when you put these fossils into a phylogeny and use that to date the phylogeny. It seems that open habitat grasses must have started diversifying at least by 55 million years or so, Paleocene, Eocene time frame. Okay, so when did these open habitat grasses become ecologically important in North America? So, and why? So, whoops, some little cut off there. Sorry about that. So the idea has long been that as climates got cooler and drier after about 50 million years or so, and drier, especially at mid to high latitudes during the Cenozoic, we see the diversification and rise in abundance of grasses, and in tandem, the evolution and diversification of large herbivores feeding on grasses. And for North America, as I mentioned, this is thought to have happened about 18 to 15 million years ago, so mid-Miocene. This is something that we've worked on testing by using phytolith assemblages extracted from Eocene through Miocene, so 40 to 5 million year old sedimentary rock samples from the US interior. And yeah, that's an example what the rock looks like. So how do we use these phytolith assemblages to reconstruct vegetation? So this is a phytolith assemblage as a pie chart. And to reconstruct habitat openness or vegetation structure, we compare phytoliths typical of open habitat grasses with phytoliths typical of woody trees and shrubs and other plants we associate with forests. And then we can look more closely at the grass communities to get some further information about the ecology and composition. This is just showing some images of what these phytoliths look like. And a general rule of thumb is that grasslands tend to have at least 50% open habitat grass phytoliths. So with that, let's look at some data. So this is showing North America, the Central Great Plains, so Nebraska, Wyoming on the left side, and Colorado on the right-hand side, and time moving forward as we go up on the y-axis. And what we can see here is that during the Eocene and Oligocene, we have assemblages dominated by forest indicators, including palms and also dominantly bamboo phytoliths. And so we reconstruct that as some kind of subtropical forest, perhaps with bamboos in the understory. But already by 34 million years ago, we see typical open habitat grasses appearing in low abundances initially. So we interpret this as these open habitat grasses maybe appearing in forest margins or glades or something like that. And then later, in the late Oligocene or early Miocene, and we do have some gaps, so where there's no rocks to sample or there's some uncertainty, and that's sort of the stippled areas there. But sometime in the late Oligocene, early Miocene timeframe, we see that these open habitat grasses are becoming ecologically more dominant and forming some kind of open grass-dominated habitats by this time. Although there's some variation in, there's still a lot of woody elements, as I'll get to in later. And importantly, these earliest grass communities were dominated by cool temperate C3 poid grasses, specifically seeming like stipod grasses, for those of you who are familiar. If we then compare this to the Northern Rocky Mountains, so Montana, Idaho, we see a similar pattern there with a shift occurring sometime in the Oligocene, Miocene, even though there are other differences. So for example, we have other close habitat grasses in Montana, and then we also have fewer palms overall and things like that. But generally speaking, this change seemed to happen roughly at the same time. We're currently doing some work in the Pacific Northwest to try to add the sort of West Coast perspective. And here we're able to look both at megaflora, so leaves, etc., pollen, phytoliths, and also cuticle. And we're also working with colleagues to compare with faunas. And here we see a very different story. So in the time frame that we have our earliest grass-dominated habitats in the continental interior, we instead see the spread of more forested habitats. So a different story on the West Coast that will be developing as we get further in our work. If we then summarize what we learn from the fossil record in North America, is that we see that the ecological expansion happened many, many millions of years after the taxonomic diversification, indicating that there were different factors driving that change. And what might those be? So, whoopsie, I went the wrong direction. Here we go. So I've done some work with colleagues, work led by Tyler Kukla, who was then at Stanford. These are geochemists, so what they were interested in using stable oxygen isotopes in clays and carbonates. And without getting into any detail, that gives you information about seasonality of rainfall. And so they looked at many measurements of these of stable oxygen isotopes across the western U.S., northwestern U.S., from the continental interior, and then they compare that to the phytolith data, showing this opening in this OHT interval, open habitat transition. And what they found was that there was a shift towards increased wintertime aridity during this same time frame, especially in the continental interior. So a working hypothesis now is that this shift in wintertime rainfall seasonality is maybe what contributed to this change, ecological change. Okay, so if we then compare this timeline for grassland evolution in the Great Plains with the evolution of Hypsodonty, we see that there was a lag of several million years. And so there was not this tight coevolution between grasses and herbivores. And so this is a little bit perplexing, perhaps. But one explanation might be that during this time frame, there was a lot of woody cover, most likely. So perhaps we had some kind of mosaic habitat with grasslands and more wooded areas. And the faunal record supports this because we see during this time a diverse mix of both browsers, so animals eating tree leaves, and grazers. And this also points to more heterogeneous vegetation during this time. Okay, let's talk now about C4 grasses. So to remind you, C4 grasses or C4 plants, C4 photosynthesis is advantageous in hot, arid climates, with summer precipitation, and also low levels of atmospheric CO2. And today, C4 grasses in the PacMed clade tend to dominate savannas and grasslands at low elevation in the tropics. As luck would have it, we can actually study the spread of C4 biomass or C4 plants, which is typically C4 grasses, using stable carbon isotopes, because the way that C4 grasses select carbon during photosynthesis is they're less selective than C3 plants, so we can get a different signal isotopically. And so people have used these stable carbon isotope records in the Great Plains, and time is here on the y-axis again, and then percent C4 on the x-axis. And what you can see there is that C4 grasslands or C4 grasses started to become important after 7 million years. So yeah, and phytolith studies have supported this time frame. So PacMed phytoliths start becoming abundant at around after 10 million years, and more so after 7 million years. And the interesting thing is that chloridoid grasses are an important part of this shift towards C4 grasses, even though we see C4 chloridoids much further back in the record, so at least 19 million years ago in North America. Also importantly, C4 grasses here replaced primarily C3 pollweed grasses, so these grasslands that I talked about earlier. Okay, so if we look at C4 grasses, then we see a similar decoupling between taxonomic diversification and ecological expansion, as we saw with open habitat grasses in general. And this again might imply that this ecological expansion depended on some other factor. And so one of these factors, sorry, let me go back, that people have talked a lot about is a drop in CO2 after the middle Miocene, which would be which would be advantageous for C4 grasses. But we also know from comparing across continents that this was not enough, and that there are many regional or continental scale factors that must have been important. And so if we look at grasses, pack-mat grasses, or C4 grasses in particular, how they respond to climate, we see today chloridoids are very strongly linked to very dry climates, whereas panicoids, for example, are linked more to a wetter climate. And so our working hypothesis is that the fact that we see an increase in chloridoids maybe indicates that there was an increased overall aridity, perhaps contributing to this change. Okay, so I hope I've shown you that the assembly of North American grasslands was a complex affair, if you will, with many different steps. Now I want to dive a little deeper into these earliest grasslands and how similar they were to what we see today. So I mentioned that they were dominated by, these grassy environments were dominated by poid grasses. Today, when we look at poids, like I said, they tend to dominate in cool and temperate grasslands. So the blue colors on the map there. And if you do a so-called ancestral state reconstruction, which is basically looking at the phylogeny of grasses to understand when different traits evolved, or in this case, climate tolerances, we see that poids overall, like early on in their evolution, they were adapted to colder and drier climates. So this is perplexing because in the early Miocene, we see them co-occurring with palms and gingers, and those are plants that we typically associate with subtropical, tropical environments and much wetter environments. So we were interested in trying to understand this a little bit more, trying to understand the ecology of these early grasslands. And so we wanted to do that looking at grass phytoliths, because they are the most taxonomically diagnostic and seem to be linked to ecology to some extent. However, the sort of traditional classification of grass phytoliths is rather coarse, also a bit subjective. So what we wanted to do was get a much more precise, robust determination of taxonomic affinity so that we could infer ecology or climate. And we did that, or we are working on doing that, by coming up with a quantitative classification using the 3D shape of grass phytoliths. And I'm going to talk about work applying so-called geometric morphometric, which is a way to measure shape. I want to acknowledge the big team of grad students, undergrads, and especially Tim Gallagher, who was a former postdoc who led this work. So what we wanted to do then was to first build a 3D classification model based on modern grass phytoliths. Then we wanted to apply that to Eocene to Miocene fossil phytoliths from the Great Plains. And then we wanted to use so-called niche models to infer climatic envelopes of grasses. And I'll talk about that a little bit later. So for this study, we sampled leaves of 43 poidy species. We added this to a data set of 50 species of early divergent grasses, bamboos, and Oryzoideae. And then we extracted phytoliths from these samples. We stained them with fluorescent dye. Then we used a so-called confocal microscope to create image stacks that we sort of sewed together to create a mesh that we cleaned and aligned and so on. And so in this, we ended up with about 2,000 3D grass phytolith meshes or models from these groups. And this is circular phylogeny showing the taxonomic coverage there. We fit landmarks, so these are points that we think are homologous or represent the same point on these phytoliths. And then we also fit 1,001 sliding surface semi-landmarks. So this is basically like throwing a net over the phytoliths. And then we transformed and aligned them to remove size, so we could compare just shape. But then we added in some size and shape variables because we think size matters. And then this is getting a bit technical now, but we created this model for classifying taxonomically the phytoliths using so-called random forest, and we did this at different taxonomic levels, so subfamily, tribe, subtribe, and so on. And then we checked how well the model did. So this is basically using the shape to try to assess where a phytolith will fit taxonomically. Okay, so to make a long story short, the model worked pretty well. So especially at the subfamily and tribe level, it worked very well. And in all cases, the accuracy was a lot better than the so-called no information rate, which is basically random guess. This ordination here is just showing how these phytoliths are separating in morphospace at the subfamily level. So hopefully you can see there's some separation. So we had our model, and then we applied that to fossils from the US Great Plains, like I said, Eocene through Miocene. We treated those fossils exactly the same way as the modern. And this is the result. So we see here on the x-axis time as we move forward, going to the left this time. And we see the fossil assemblages where we were able to classify these grass phytoliths. And what we can see here is that poet grasses were present much earlier in the Eocene than we previously thought, if you want to trust this model. And also, we were able to refine some taxonomic inferences. For example, we were able to show that most likely we had some of the Poe subtribes present in these earliest grasslands. However, there's a lot of work to be done to expand our data set, and also we're applying some different approaches. Yeah. So then we wanted to infer climate, like I said, and we did this in two different ways. One is a so-called nearest living relative approach. So that's basically, like I said, we reconstructed the climate preference or tolerance across the phylogeny. So on the left-hand side, it's mean annual temperature. On the right-hand side, it's mean annual precipitation. So we see those evolutionary patterns. And then we place our fossils in the phylogeny as best we can using our model. And then we see what climate that seems to represent. That method is a little bit imprecise at the moment. So we mostly relied on an ataxonomic approach that we developed, which was looking at the correlation between phytolith shape and these two climate parameters. And what we found when we used that approach is that, just let me walk you through this. So here we're also showing data from Turkey. You can just ignore that. But basically on the left-hand side, we have a mean annual temperature reconstruction. On the right-hand side, mean annual precipitation. And the blue line is the current temperature in North America and precipitation. And as you can see, even though we have a lot of uncertainty, climates were overall warmer and wetter back in the Miocene than Oligomycene than we have today in the Great Plains, also in Turkey. But and that reconstruction is consistent with the presence of palms at all of our sites. We had in a previous study looked at palm phytoliths and determined that most likely the palms we see in the Great Plains back then were warm and wet adapted. So again, this just shows again that this was a rather non-analog ecosystem. So in conclusion, I just wanted to say that, in conclusion, grassland evolution in North America occurred in several steps. First, we see the expansion of these C3 dominated grassy habitats, mosaic. That's in the Miocene there, the yellow. And then we later see the expansion of C4 grasslands and a much more open habitats after 7 million years or so, and even more so after 5. Also, the earliest grasslands were not like any grasslands we have or grassy environments that we have around today in North America and elsewhere. And so, yeah, so this is curious, but it's also really important to understand how these early grasslands functioned. Because that gives us insights into how grasslands function today and how they might change in the future as we are moving into climates that are potentially non-analog. And just as a sort of an example of how the evolutionary history of grasslands are important for informing current and future function, this work has been able to help show that grasslands are not just what happens when you cut down trees. They are ancient ecosystems in their own right with a unique biodiversity, and they deserve to be protected. Thank you for your attention.